Hello everyone, now let us discuss about Good Manufacturing Practice Guide for Active Pharmaceutical Ingredients Part 12. This comes under Q7 Quality Guideline of ICH. In this session, we will be discussing the specific guidance for API manufactured by cell culture or fermentation. Specific guidance for APIs that are manufactured by cell culture or fermentation. Now, coming to general guidelines. They are intended to address specific controls for APIs or intermediates manufactured by cell culture or fermentation using natural or recombinant organisms that have not been covered adequately in the previous sections. It is important to note that principles of fermentation for classic processes for the production of small molecules and for the processes using recombinant and non-recombinant organisms for the production of proteins and or polypeptides are same, although the degree of control will differ. And where practical, this session will address these differences. In general, the degree of control for biotechnological processes used to produce proteins and polypeptides is greater than the classic fermentation process. The term biotechnological processes refers to the use of cells or organisms that have been generated or modified by recombinant DNA, hybridoma, or other technology to produce APIs. The APIs produced by biotechnological process normally consist of high molecular weight substances such as proteins and polypeptides, for which specific guidance is given in this session. Certain APIs of low molecular weight such as antibiotics, amino acids, vitamins and carbohydrates can also be produced by recombinant DNA technology. And the level of control for these types of APIs is similar to that of employed for classical fermentation. The term classical fermentation refers to the process that uses microorganisms existing in the nature and or modified by conventional methods, for example irradiation or chemical mutagenesis, to produce APIs. The APIs produced by classical fermentation are normally low molecular weight products such as antibiotics, amino acids, vitamins and carbohydrates. And the production of APIs or intermediates from cell culture or fermentation involves biological processes such as cultivation of cells or extraction and purification of material from living organisms. Note that there may be additional process steps such as physicochemical modifications that are a part of manufacturing process. And the raw materials used, for example media, buffer components, may provide the potential for the growth of microbial contaminants. Depending upon the source, method of preparation and intended use of the API or intermediate, control of bioburden, viral contamination and or endotoxins during the manufacturing and monitoring the process at appropriate stages may be necessary. Proper control and monitoring of the process at appropriate stages is necessary. Appropriate control should be established at all stages of manufacturing to assure intermediate and or API quality. While this guide starts at the cell culture or fermentation step, prior steps, for example cell banking, should be performed under appropriate process controls. And this guide covers cell culture or fermentation from the point at which a vial of the cell bank is retrieved from the further use in manufacturing. Appropriate equipment and environmental control should be used to minimize the risk of contamination. The acceptance criteria for the quality of the environment and the frequency of monitoring should depend on the step in production and the production conditions, for example open, closed or contained systems. The acceptance criteria for quality should depend upon the step in the production and the production conditions. In general, the process control should take into account the first one is maintenance of the working cell bank where appropriate, proper inoculation and expansion of the culture, control of the critical operating parameters during fermentation or cell culture, monitoring of the process for cell growth viability for most cell culture process and productivity where appropriate, harvest and purification procedures that remove the cells, cellular debris and media components while protecting the intermediates or API from the contamination and from the loss of quality. monitoring of bioburden and, where needed, endotoxin levels at appropriate stages of production, viral safety concerns. The viral safety concerns are described in ICH Guideline Q5A. Viral safety concerns are described in ICH Guideline Q5A. Quality of biotechnological products. Viral safety evaluation of biotechnology products derived from cell lines of human or animal origin. And where appropriate, the removal of media components, host cell proteins and other process related impurities, product related impurities and contaminants should be demonstrated. Coming to cell bank maintenance and record keeping. Access to cell banks should be limited to authorized persons. Cell banks should be maintained under storage conditions designed to maintain viability and prevent contamination. Records of the use of the vials from the cell banks and storage conditions should be maintained. Wherever appropriate, cell banks should be periodically monitored to determine the suitability for use. Coming to cell culture and fermentation, where aseptic addition of cell substrate, media, buffers and gases is needed. Closed or contained systems should be used where possible. If the inoculation of the initial vessel or subsequent transfers or additions, for example media or buffer, are performed in open vessels, there should be controls and procedures in the place to minimize the risk of contamination. Whenever aseptic conditions are needed, it is better to perform in closed or contained systems. But whenever open vessels are used, there should be controls and procedures in place to minimize the risk of contamination. Where the quality of the API can be affected by microbial contamination, manipulations using open vessels should be performed in a biosafety cabinet or similarly controlled environment. The personnel should be appropriately gowned and take special precautions while handling the cultures. Critical operating parameters. For example, temperature, pH, agitation rates, addition of gases, pressure should be monitored to ensure consistency with the established process. Cell growth, viability for most cell culture process, and where appropriate, productivity should also be monitored. Critical parameters will vary from one process to another, and for classical fermentation, certain parameters, for example, cell viability, may not be monitored. Cell culture equipment should be cleaned and sterilized after the use. As appropriate, fermentation equipment should be cleaned and sanitized or sterilized. Culture media should be sterilized before use when appropriate to protect the quality of the API. There should be appropriate procedures in place to detect the contamination and determine the course of action to be taken. And this should include procedures to determine the impact of the contamination on the product and those to decontaminate the equipment and return it to a condition to be used in subsequent batches. Foreign organisms observed during fermentation process should be identified as appropriate, and the effect of their presence on the product quality should also be assessed if necessary. The results of such assessment should be taken into consideration in the disposition of the material produced. Coming to records of contaminations. Records of contamination events should also be maintained. Shared equipment, for example, multi-product, may warrant additional testing after cleaning between product compounds as appropriate to minimize the risk of cross-contamination. Whenever shared equipment is used, additional testing after cleaning between product to product is necessary to minimize the risk of cross-contamination. Now coming to harvesting, isolation and purification. Harvesting steps either to remove cells or cellular components or to collect cellular components after disruption should be performed in equipment and areas designed to minimize the risk of contamination. Harvest and purification procedures that remove or inactivate the producing organisms, cellular debris and media components while minimizing degradation, contamination and loss of quality should be adequate to ensure that the intermediate or API is recovered with consistent quality. All the equipment should be properly cleaned as appropriate, sanitized after use. Multiple successive batching without cleaning can be used if intermediate or API is not compromised. If the intermediate or API quality is not compromised, you can use multiple successive batching without cleaning. This is mostly applicable for only one product equipment. If the equipment is used only for one product, you can use multiple successive batching without cleaning, provided if the intermediate or API quality is not compromised. If open systems are used, purification should be performed under environmental conditions appropriate for the preservation of product quality. Additional controls such as the use of dedicated chromatography resins or additional testing may be appropriate if the equipment is to be used for multiple products. Coming to viral removal or inactivation steps. Viral removal and viral inactivation steps are critical process steps for some processes and should be performed within their validated parameters. Appropriate precautions should be taken to prevent potential viral contamination from pre-viral to post-viral removal or inactivation steps. Therefore, open processing should be performed in areas that are separate from the other processing activities and have separate air handling units in order to control the microbial count in the environment. The same equipment is not normally used for different purification steps. However, if the same equipment is used, the equipment should be appropriately cleaned and sanitized before reuse. After, appropriate precautions should be taken to prevent the potential virus carryover. Example, through equipment or environment from previous steps. Appropriate precautions should be taken to prevent the potential virus carryover from the previous steps. Thank you for watching. Please subscribe for further videos on pharmaceutical sciences and other related disciplines.